Hello, everyone. Today we're diving into the intricate world of RNA processing, specifically focusing on two crucial mechanisms, RNA capping and polyadenylation. These processes ensure the integrity and stability of mRNA, which is fundamental for proper cellular function. First, let's review the basics. RNA, or ribonucleic acid, is a crucial player in the cellular orchestra. It's transcribed from DNA and serves as a messenger, carrying genetic instructions for protein synthesis. But to perform its role effectively, RNA needs to undergo a series of modifications. These two processes are like giving our RNA a protective shield. We'll start at the 5' end with 5' capping. This process occurs co-transcriptionally. Typically after 20 to 100 nucleotides are synthesized by RNA polymerase. Now, let's break down the steps involved. Phosphate is at the 5' end of each nucleotide. The first step involves removal of a 5' phosphate of RNA by RNA triphosphatase. Next, guanosine triphosphate undergoes hydrolysis, and so guanosine monophosphate is added to the 5' diphosphate of RNA by RNA guanylyl transferase. Then, methyl groups are transferred via methyltransferase. The methyl group is donated by S-adenosylmethionine, or SAM. And then it's brought by methyltransferase to the N7 of this blocking guanine. Finally, another similar transfer occurs. This time, it's brought to the hydroxyl group of the first and second nucleotides, and thus forms the 5' cap. So, why is capping so crucial? The 5' cap acts like a protective armor, resembling the 3' end of RNA, so that provides significant resistance against 5' exonucleases. It also interacts with cap binding complexes CPB80 and CPB20, as well as cap binding complexes EIF4E and EIF4G. These are nuclear and cytoplasmic, respectively. The latter enhances translatability by attracting translation initiation factors and facilitating the beginning of translation. It's also required for mRNA export out of the nucleus and promotes splicing of the 5' most intron. So basically, capping increases the likelihood of translation initiation and it protects against degradation. Now, let's shift our focus to polyadenylation, the process of adding a poly A tail to an RNA transcript. Polyadenylation begins with the recognition of a specific signal, often the AAU-AAA sequence. It's located upstream of the cleavage site. This is a very simplified description of the machinery. There are many additional components. RNA polymerase II acts as a landing pad for the following polyadenylation machinery. Once the signal is identified, cleavage and polyadenylation specificity factor, or CPSF, is recruited to the mRNA molecule. Then, cleavage stimulatory factor recognizes and is situated by the GU-rich region. Cleavage factors 1 and 2, depicted here as scissors, help the CPSF cleave the mRNA. The cleaved 3' RNA is rapidly degraded. Poly A polymerase is activated. With very high specificity, it uses ATP and magnesium ions to build a poly A tail. It will synthesize a short A oligo, one residue at a time, and thus completes poly A tail synthesis initiation. The second and final step in the process is poly A tail elongation. Elongation requires poly A binding protein 2. It binds the short oligo A pre-initiated by PAP. These three work in concert to rapidly make a poly A tail about 250 nucleotides in length. Once matured, these polyadenylated mRNAs exit the nucleus to enter the cytoplasm, where translation will occur. Once in cytoplasm, these tails are bound by PAB1. If you guessed that it helps protect the mRNA from degradation, you're correct. It also facilitates translation by binding with the aforementioned EIF4G. These interactions create a loop of the 5' mRNA cap and the 3' mRNA poly A tail. It's hypothesized that this enhances translation. And just like the mRNA in the cytoplasm with its 5' cap and its 3' poly A tail, this story has come full circle. Until next time, keep exploring the wonders of molecular genetics.